Hello, everyone. I'm David Carey of FINN Partners, and joining me today is Dr. Juho Jalkanen, CEO of Faron Pharmaceuticals. Faron is a clinical stage biopharmaceutical company at the forefront of a clever approach to cancer treatment. By focusing on the role of the myeloid system in the tumor microenvironment, Faron is pioneering first-in-class immunotherapies designed to reprogram the immune system to recognize and attack cancer cells. Their lead asset, bexmarilimab, is a novel anti-Clever-1 antibody that targets a key immunosuppressive pathway, potentially turning cold tumors hot and overcoming resistance to existing standard of care treatments. Juho joins us at a pivotal moment for Faron, following highly encouraging clinical data presented late last year at ASH 2025. The company's flagship BEXMAB trial investigating bexmarilimab in combination with azacitidine has shown remarkable efficacy in patients with HRMDS, a population that has seen little innovation in decades. With phase 2 enrollment complete and a clear registrational pathway aligned with the FDA, Faron is now moving forward with a phase 2/3 trial. And beyond these successes in hematological malignancies, Faron is also expanding its reach into solid tumors. The company recently announced a strategic collaboration with the Institute of Cancer Research to launch the BLAZE trial. This is a phase 1/2 study which will investigate bexmarilimab's ability to overcome acquired resistance to checkpoint inhibitors in patients with lung cancer and melanoma, seeking to prove the drug's potential to resensitize tumors to immunotherapy. So, to start out, Juho, in your words, can you please tell us a little bit more about Faron and bexmarilimab?
Well, thanks, David. I'll be happy to. Good to meet everybody. Faron is a publicly listed company both on the Helsinki and London stock exchanges, and we will be making forward-looking statements in this presentation. So we've been working 20 years in the field of tumor immunology, really looking to break treatment resistance. How could we make existing treatments work better for patients? Really drilling down to what's happening in the tumor biology. So that's where we originate from, and looks like we might have struck gold here. And we're laser focused in high risk MDS, as you said. That's myelodysplastic syndrome. It's a rare and deadly form of leukemia, so blood cancer. And it's where the macrophages have gone wild, and the macrophages are the cancer themselves. So that's where we really seem to work, and there's been no new innovation in this field. So if we look at the first slide of innovation highlights, laser focused on delivering finally something new for these dire patients. To do that, we use our first-in-class anti-Clever-1 antibody, bexmarilimab. We're going to call it again Bex from here on because it's a lot easier. As you mentioned, we just completed a phase 1/2 trial, presented at ASH very recently, and we've also held an FDA meeting in the phase 2 meeting that what would a registrational trial look like, and that's what we're gonna step into next. So super exciting times for MDS patients and us for the company. But the story doesn't, as you also mentioned, doesn't end with MDS. This is possibly highly applicable to a number of cancers where macrophages are the source of treatment resistance. So this is actually then the next slide on what does our pipeline look like. So in purple there is the flagship MDS program, and the registrational trial there is named BexNova. We've also in orange here worked a lot in solid tumors. And through this first in human solid tumor trial, which was named MATINS, we've now designed a number of also solid tumor trials in blue, which will be starting. These are again phase 1, 2 proof of concept trials that we could overcome treatment resistance in a number of very hard to treat cancers. As you mentioned, checkpoint refractory lung and melanoma, then also sarcomas, where again nothing seems to work. It's a bit like MDS, that nothing seems to work. So we really wish to show the world that it seems to be that we're on to something super special, that's something that could really overcome treatment resistance. So I would also like to spend a little bit of time on the actual science and how does this drug work. So this next slide illustrates the mode of action. So Clever-1, the target, it's a scavenger receptor on macrophages and monocytes. The purpose of these cells as a part of the immune system is to go around, clean up, eat debris, dying cells, and foreign matter like cancer and present that to the immune system. So, and they actually activate the T cells that then kill cancer and foreign material like bacteria. So, but it needs the monocytes and macrophages to show them what is foreign. And the problem with these Clever-1 positive macrophages is whatever they eat, they don't present. But when you target them with BEX, instead of hiding the tumor from the immune system, it starts presenting the tumor to the immune system again and makes the immune system attack cancer. Now, in the context of MDS, which is a very difficult to treat cancer, the actual cancer cell is a macrophage. It's a juvenile gone wild form called the blast cell, which you can see in this slide. So they're equipped with their own protective element through Clever-1. So now why we're possibly seeing these excellent results is we're not only activating the immune system, we're also depriving the cancer of its main source of protection.
That's great, Juho. Thank you. So I think this is a good segue into MDS and especially HRMDS. Can you tell us a little bit more about the disease and why it is so difficult to treat?
Yes, no, that's a super good question. And possibly this biology we're seeing is one of the core reasons why that nothing seems to work because it comes with such a good protective agent, Clever-1, and we're going to attack it. So this is possibly one of the first drugs to actually tackle the core biology of higher risk MDS. Now, why MDS is so difficult and a dreadful disease is that again, it's a form of leukemia. So the problem is this is really deep in your bone marrow where blood cells are produced. And the bone marrow isn't producing any more healthy blood cells like red blood cells, white blood cells. It's only producing these cancerous blast cells. So these patients become deeply anemic, neutropenic. They suffer from anemia, constant infections. They're in and out of the hospital, getting blood transfusions, IV antibiotics. So ultimately, the quality of life deteriorates, and then also your prognosis becomes very poor. So actually, with high risk MDS patients, usually after diagnosis, they only live 8 to 24 months. And if you're treatment resistant, as many patients are originally or become, then nothing really seems to work, and survival is only a mere 5 to 6 months. So we're talking about a very deadly form of leukemia here. Currently, these patients are treated with a form of chemotherapy called hypomethylating agents, HMA, and the market leader is azacitidine, as you mentioned. We can call it AZA to make it again easier for the audience. So the frontline and basically only treatments are these HMAs. Problem with them is 50% do not respond at all, and then the majority eventually relapse. And again, there's really no approved treatment for the general population after that. You can then try conventional chemos, but nothing really seems to work when you're so-called last line or relapse refractory MDS. So that's why it's so difficult and desperately new treatment options are needed.
Yes, indeed. So, can you tell us a little bit about what Farron has been doing in the field, and what do some of your latest results tell us?
Yes, so we've been running this BEXMAP phase 1/2 trial. It's been running in the US and in Europe. Uh, where leading sites, MD Anderson, City of Hope, Yale, and so forth, have been working with us on this. And we've been treating both frontline high-risk MDS patients and last-line high-risk MDS patients. And what we're seeing now in this slide is the results from the frontline population. And CR is a complete remission. Uh, Cancer totally gone away, blood counts to normal. ORR is the objective response rate here in the left column. Usually with azacitidine as the first treatment, you would see a CR rate of 16 to 17%. We're at 45. And the again objective response rate usually is around 40-45%. We are at 85. And on the right hand side here you see a so-called waterfall plot. That's the blast cell count, so counting the amount of cancer cells in the bone marrow, and you see the majority of patients go to zero. Basically everybody seems to be responding, so these are extremely encouraging results, again, in the frontline setting together with azacitidine. What's even more striking is that TP53, which we're reporting here, is a very bad mutation to have. You know, cancers are mutation driven. It's a very bad mutation. Nobody really seems to respond when you're TP53 mutated. We seem to work extremely well also in this very hard to treat population. The CR rate was 70% in this TP53 mutated population, which is basically unheard of before. Uh, but even more encouraging than this is the last line population, which we're seeing here in the next slide. So this is the population that has exhausted all resources, nothing available. You can try re-challenging with an HMA or chemotherapy. You can expect 0 to 20% response rate. We're at 64. So again, the majority are now suddenly again responsive to azacitidine. And this patient data set is so-called more mature. And as mentioned, usually this patient population, usually their survival is around 5 to 6 months. Super encouraging is the survival, which we're showing here in this so-called Kaplan-Meier survival curve, is that we've taken that standard 5 to 6 months to 14.5 months. Again, this is the best survival ever reported in such a population. Here in the graph, we've separated the TP53 mutated and not. For the TP53 mutated, the OS, which is the overall survival, usually is only 3 to 4 months. We're at 7.5, and again for the entire population, 14.5. So this has been extremely encouraging for the MDS field.
The efficacy looks great, Juho, but what about the safety in this otherwise frail and anemic population?
Well, that's the best thing about actually this drug. So we're not a cytotoxic cancer killing agent as such as, traditional cancer treatments are usually toxic because they're meant to kill cancer. We are an immune modulating, immune system reprogramming agent. So here in this slide, this is how our safety compares to other drugs that have been recently trialed in high-risk MDS. So in orange is the safety for Bex, and in gray, the safety for the recently, unfortunately, failed programs, and a lot of these hematological parameters. So the problem with this extremely frail, anemic, neutropenic population is that azacitidine, being a form of chemo, actually usually makes the anemia and neutropenia even worse. And then if you add on another toxic agent, that's basically going to break the camel's back, and this patient population just cannot stand another toxic treatment. And this is how we stand out as not being a cytotoxic treatment, but a true immunomodulating disease modifying agent that we don't make anemia and neutropenia worse. We actually seem to make it better. And that is, I think, key to success in this otherwise very difficult patient population.
Okay, so what happens now in development?
So we just also reported these excellent results, but met with the FDA. And what we designed with the FDA's advice is this phase 2/3 program. So FDA still wanted us to look at the dose a bit in more detail. So we planned this phase 2 trial where we start with still 2 doses of Bex because the program is advancing pretty fast. We're coming out straight from that, moving to this. We still need to advance the program rapidly for these patients, but we still need to do a bit of work on the dose. So that's why we have 2 doses in blue here in this slide. And then the inferior dose drops away, and seamlessly these other patients move on to the final phase 3 stage, and we add on patients. And now the great news from the FDA meeting was that FDA will accept from this one single approach that there's possibility for first accelerated approval. So pretty early on in the final program. Usually these phase 3s, take a long time, but here we may be able to achieve accelerated approval based on the response rate, again, the CR rate of getting patients into remission. Now, in cancer trials nowadays, overall survival is the ultimate endpoint. You want to see a survival benefit. So that will be a second endpoint that will then be used as the final readout and the full approval of the drug later on. But to make this drug available earlier for patients, there's the accelerated approval possibility basically halfway through the trial.
Okay, great. So why don't we shift over to solid tumors then? And you mentioned that, you know, this could work in many different types of cancer. So what's the reasoning and approach towards solid tumors at Farron?
Yes, so a lot of in solid tumors, actually, 50% of the tumor mass is usually made of macrophages. And what we've seen is, screening a lot of cancers is that in these treatment-resistant solid tumors, they're usually filled with these clever one positive macrophages. Like in a sense, MDS is. It's all clever positive. But in these very nasty solid tumors, you find these clever positive macrophages. So again, they're probably a key source of treatment resistance. We are most likely to be able to overcome that. So what we've actually already learned in solid tumors, and this comes from our initial first in human trial in solid tumors, is PD-1s, when they came, great drugs, they revolutionized, immunotherapy in cancer, helped out a lot of melanoma and lung cancer patients. The problem is still Uh, Quite a lot of patients don't respond to PD-1s or relapse on them. It's very well known that for PD-1s to work, they basically unleash the T cells. So you want an ongoing immune reaction in the tumor. You can measure that using a pro-inflammatory cytokine named interferon gamma. So PD-1s usually work in this interferon gamma high population. And in the left-hand picture, again, which is a Kaplan-Meier survival curve, these are melanoma patients treated with nivolumab, so a classical anti-PD-1. And the survival benefit, the clinical benefit is seen in the interferon gamma high population. Now, on the right-hand side are single-agent Bex-treated melanoma patients from the actual first-in-human study. And for us, it's the opposite. We're going to actually significantly benefit the interferon gamma low population. And interferon gamma low is the so-called cold tumor, non-responsive immunosuppressive cold tumor, where nothing really seems to work. So we're going to actually ignite those immune responses. We're going to elevate interferon gamma in the tumor microenvironment. We truly believe that we're going to make PD-1s work in patients where they didn't work before. So, and if this holds true and this trial is now starting, this is going to be big.
Hey, great. Well, Juho, any final thoughts before we wrap up?
Well, I'm and all of us here in Faron are super excited about this program. So are a lot of investigators, physicians we work with. Uh, Come and join, come follow us. Amazing times ahead, hopefully for all cancer patients.
Great, Juho. Well, I want to thank you for participating today in the BioInnovation Forum. Really appreciate it. I know that you're actually right now in San Francisco, and you're doing meetings surrounding the JP Morgan conference. So hopefully you're having a productive week out there, and that looking forward to hearing more about Farron in the future.
Yes, thanks, David. Running up and down these hills here, it keeps you fit at least, but enjoying
Definitely,
myself.
definitely. Well, have a good rest of the conference.
Thank you.